Now let's go to the inner ear. So the inner ear is contained in the pectus apex of your temporal bone and is encased in a bony structure called your osseous labyrinth or your bony labyrinth. So the bone that forms the cochlea and vestibular labyrinth is the hardest bone in the human body and is akin to ivory in its density. The labyrinth consists of 3 continuous sections. You have the vestibule, the cochlea, and your semicircular canals. The initial point of communication between the middle and inner ear occurs at the oval window of the vestibule, where the stapes footplate abuts the oval window membrane. So remember meso, so from your malleus, incus, stapes, and then oval window. So at the basal end of the cochlea is the round window membrane, which communicates with the middle ear space. Okay, so here, in purple, that's your bony labyrinth. And then in blue, that's your membranous labyrinth. Okay. So let's first talk about the cochlea. So the cochlea is a snail-shaped structure that has a wide diameter at the base, which narrows for 2 1/4 turns until it reaches its apex. So this picture shows a cross-section of the cochlea and illustrates the cuts through each turn. So the core of the cochlea is the modiolus. Okay, so there. A highly porous bone that allows passage of auditory nerve fibers that travel from the internal auditory meatus to the hair cell synapse. So extending from the modiolus into the osseous labyrinth space is a bony shelf, or you have your osseous spiral lamina. So this coils around the center of the cochlea and provides partial division of the upper and lower cochlear chambers into the scala vestibuli and scala tympani. So at the apex of the cochlea, these scala or scalae communicate at your helicotrema. Okay, so here, there. So the spiral lamina is also the point of attachment for the basilar membrane, which is the lower border of the membranous labyrinth that encases the scala media. So along the length of the cochlea, the widths of the spiral lamina and basilar membrane are inversely related, with the spiral lamina wider at the base and narrowing towards the apex at the basilar membrane, and narrower at the base and wider at the apex. So this is one of several contributing factors to the frequency specificity of basilar membrane motion. So at the basal turn of the cochlea is your cochlear aqueduct. Okay, so which is a bony channel that allows communication between your perilymphatic fluid and cerebrospinal fluid of the subarachnoid space in your posterior fossa. Okay, we'll go through that in more detail. Then that's your helicotrema. That's your basilar membrane. Okay. So the membranous labyrinth of the cochlea follows the shape of the osseous cochlea and forms a third cochlear chamber, which is your scala media. So the sensory organ of hearing resides within the membranous labyrinth. So this figure shows a cross-section of the membranous labyrinth and its structures. So this labyrinth is bordered superiorly by your Reisner's membrane, here, inferiorly by your basilar membrane, there, okay, and laterally by a portion of the outer cochlear wall to which it is anchored by your spiral ligaments. This is the lateral, okay. So within the membranous labyrinth, along the lateral wall is your stria vascularis. So the stria vascularis is a highly vascular tissue that is responsible for the metabolic environment of the scala media. The intricate structure that comprise the organ of Corti are situated on the basilar membrane. So dito sa baba. Okay. So the organ of Corti runs longitudinally along the length of your basilar membrane and consists of many types of epithelial cells and structures. So medially seated atop the osseous spiral lamina is your spiral. So this is your spiral lamina. Ito, this one. So, on top of that is your spiral limbus. Okay. So, a thickened band of periosteum that serves as the point of medial attachment of your Reisner's membrane. So, dito naka-attach yan, diba? This is your Reisner's membrane. There. Okay, and gives rise to the tectorial membrane, which lies over the inner and outer hair cells. So, this is your tectorial membrane. So, the tectorial membrane is a compliant gelatinous structure composed primarily of collagen II fibers, and it serves as a mass load that moves similar to a rubber band. Lateral to the spinal limbus is the inner spiral sulcus, which lined with border cells of held. Okay. One row of inner hair cells is present, and the cell bodies are surrounded by supporting cells called your phalangeal cells. So here, this is your inner hair cell. Okay. Tapos you have your outer hair cells here. Okay, so between the inner and outer hair cells lie your pillars of Corti, which originate on the spiral lamina and basilar membrane and converge at top to form your tunnel of Corti. Lateral to this are three rows of outer hair cells, which are securely cupped inferiorly by supporting diater cells. So, ito si diater cells. Yeah. Okay. So each diater cell has a phalangeal process that projects apically, and the space between the outer hair cell and phalangeal process is referred to as the null space. So the phalangeal processes of the diater cells and superior surface of the hair cells form your reticular lamina, which is a tightly interwoven matrix that supports the apices of the hair cells. The reticular lamina forms a barrier from your endolymph, the fluid in the scala media, which, owing to its ionic composition, is toxic to the hair cells. Lateral to the outer hair cells are your Hensen cells and Claudius cells. So the two fluid systems within the cochlea create an environment crucial to the mechanical displacement of basilar membrane traveling wave and to the cellular depolarization and subsequent synaptic activity. Between the osseous and membranous labyrinth is your perilymphatic fluid or your perilymph, which has a high concentration of sodium and a low concentration of potassium, similar to what is found in your cerebrospinal fluid and your blood serum. The presence of perilymph in the intercellular space within the organ of Corti has been identified in humans and guinea pigs. So within the membranous labyrinth is your endolymphatic fluid, or what we call your endolymph, with a high concentration of potassium and low concentration of sodium, such as is typically found intracellularly. Okay, so the ionic concentrations of endolymph are maintained by the cells within your stria vascularis. So the endolymphatic sac communicates with the membranous labyrinth via endolymphatic duct and vestibular aqueduct. So disorders of the endolymphatic system that results from diseases, congenital deformities, or other insults can lead to potentially severe auditory and vestibular symptoms. So an enlarged vestibular aqueduct, defined as a diameter larger than 1.5 millimeter at the vestibular aqueduct midpoint or greater than 2 millimeter at the operculum, may lead to sudden and progressive sensorineural hearing loss. Okay. Here. So for the hair cells, so the inner and outer hair cells function as receptor cells that transduce mechanical movements into an electrochemical signal to stimulate your auditory nerve. So here, this is a schematic, the picture presenting examples of inner ear and outer hair cells. So the apical portion of all hair cells include a thickened region called the cuticular plate. Okay, which in conjunction with the supporting cells forms your reticular lamina. So here, this is your cuticular plate. Okay? So, rooted in the cuticular plate of each hair cell and projecting through the reticular lamina are bundles of actin filaments, known as your stereocilia. Stiff hair-like structures that deflect with mechanical disturbances. So, these are your stereocilia. So, adjacent to this is a non-cuticular region that contains a rudimentary kinocilium. Approximately 3,500 inner hair cells are arranged in one row on the modular side of the tunnel of Corti, and 12,000 outer hair cells fall in 3 rows on the strial side. The stereocilia on outer hair cells form a V or W shape with the bottom facing away from the modulus. The inner hair cell stereocilia forms a shallow U, okay, that opens towards the modulus. So on each hair cell are rows of stereocilia, 2 or more on inner cells and 3 or more on outer hair cells. For both types of hair cells, the stereocilia lengths are graduated. They are longest on the strial side and are shortest on the modulus side. The longest stereocilia on the outer hair cells contact the tectorial membrane, which results in deflection of the stereocilia with basilar membrane movement. The stereocilia are connected to each other by filamentous links laterally by cross links, and from the tips of shorter stereocilia to the sides of taller ones by tip links. Now, these ensure that the connected stereocilia move as a unit when the longer stereocilia are deflected. The structural aspects of the inner and outer hair cell bodies differ significantly and are reflective of their functional differences. The inner hair cells are flask-shaped, wide at the bottom, and narrow at the top, and contain high concentration of organelles that are involved in metabolic activity, particularly your Golgi bodies, so here, and your mitochondria. There. Okay. Although highly metabolic, inner hair cells are considered to be passive transducers in the auditory system. Outer hair cells are cylindrical. So here, cylindrical siya. So these are your outer hair cells. So they are cylindrical and contain microfilaments and microtubules along the length of the cells that give rise to motile activity. These motile properties have been shown empirically to result in highly tuned frequency-specific contractile activity even when stimulated in isolation from the basilar membrane. All hair cells have synaptic bars at afferent synapses that serve as sites for presynaptic vesicle docking and release for subsequent stimulation of auditory nerve fibers. So in humans, afferent innervation of the cochlear hair cells consists of approximately 30,000 auditory nerve fibers, and these are responsible for providing ascending information from the cochlea to the central auditory system. The cell bodies of afferent fibers make up the spiral ganglion that reside in the Rosenthal canal within the modulus. Nerve fibers reach the hair cells by traveling through the modulus and into the osseous spiral lamina, where they pass through the holes in the lamina, referred to as your habenulae perforata. The nerve fibers are classified as type 1 and type 2, and the type 1 fibers are bipolar of large diameter and are myelinated, and they comprise nearly 95% of the fibers. Each type 1 fiber has a direct and independent synapse on the body of a single inner cell, and each inner hair cell is innervated by approximately 20 such fibers. So the type 2 fibers constitute the remaining 5%, and they are smaller and may be myelinated or unmyelinated. Type 2 fibers synapse directly on the outer hair cells, and a single fiber diverges to form branches that synapse with multiple other outer hair cells. The afferent auditory pathway originates from the olivocochlear bundle and provides central inhibitory modulation of hair cell activity via descending information. The olivocochlear bundle has about 1,600 fibers, and these constitute the uncrossed olivocochlear bundle and crossed olivocochlear bundle. These pathways originate from the medial superior olive and lateral superior olive regions on both sides. For the uncrossed olivocochlear bundle, the LSO, or your lateral superior olive, projects many small diameter unmyelinated efferent fibers towards the ipsilateral inner hair cells, where they synapse with the afferent fibers. The MSO projects fewer myelinated fibers in ipsilaterally that synapse directly on the outer hair cells. For the crossed olivocochlear bundle, the MSO projects large diameter myelinated fibers to the contralateral outer hair cells, and the LSO projects a few unmyelinated fibers to the contralateral inner hair cell afferents. So the fibers from the crossed olivocochlear bundle cross midline at the level of your fourth ventricle. Okay. Okay. Okay, now let's go to the central auditory pathway. So the central auditory pathway involves all ascending and descending neuronal projections that interconnect the auditory nerve, brainstem, midbrain, thalamus, and cerebral cortex. So one way to simplify things, no, just remember E. coli. So first, the E, you have your eighth nerve or your cochlear nerve. So the cochlear nerve transmits auditory information from your inner and outer hair cells to your brainstem. Then you have your cochlear nucleus. So all peripheral auditory information enters the central auditory system through the cochlear nucleus. So within the cochlear nucleus, initial auditory processing occurs and fibers are distributed to higher brainstem centers. So the dorsal, so have a dorsal and ventral. So yung dorsal cochlear nucleus is typically laminar in organization and is more akin to the structure of the cerebellum than to the other brainstem nuclei. It is less robust in humans than in other mammals, which typically have mobile pinna, and the dorsal cochlear nucleus may play a role in orienting to a sound source. The dorsal cochlear nucleus has also been implicated in several studies as a potential site for generation of tinnitus, or ringing sound sa ear. So the dorsal cochlear nucleus is tonotopically organized, with low frequency represented ventrolaterally and higher frequencies represented dorsomedially. Okay. Then you also have the ventral cochlear nucleus. So the ventral cochlear nucleus is divided into anterior ventral and posterior ventral subnuclei, each with an individual tonotopic map. These subnuclei have unique and shared neuronal subclasses that serve as the initial processors of auditory nerve information, prominent spherical bushy cells. Characteristics of the anterior ventral nucleus send large caliber axons to the olivary complex and function in sound localization. The multipolar or stellate neurons associated most with the posterior ventral cochlear nucleus send multiple fine axons to several other brainstem centers, including the contralateral cochlear nucleus, the ipsilateral dorsal cochlear nucleus, and the contralateral inferior colliculus. These cells likely play a role in encoding frequency, spectral shape, and sound intensity. Okay, next you have our superior olivary complex. So, superior olivary complex is the first central auditory center to receive binaural innervation, and it maintains the tonotopic organization seen in the cochlea and cochlear nucleus. Binaural innervation is indicative of the major role this nucleus plays in localizing sounds in space. So this is very important, no? So the first central auditory center to receive binaural innervation, superior olivary complex. Sorry. Okay, let's go back. Okay, so segregated cortical pathways also seem to exist for the perception of musical elements such as melody and timbre that are analogous to speech, and the interpretation of verbal content versus recognition of the speaker's voice. Reciprocal projections exist between the auditory cortex and lower auditory nuclei. Although not definitively identified in humans, three principal descending pathways to the thalamus, midbrain, and brainstem have been reported. So the primary auditory cortex projects to the other cortical regions and to the medial geniculate body. Projections from cortex to your SOC and inferior colliculus seem to contact neurons that feed back to higher centers. Direct cortical projections to cochlear nuclei also have been reported in mammalian models, and these pathways may enable the cortex to modulate ascending auditory input. Okay, so again, E. coli. Okay, so your auditory nerve or cochlear nerve. Then you have your cochlear nucleus, your dorsal and your ventral. Okay, then you have your superior olivary complex. Then you have your lateral lemniscal nuclei or lateral lemniscus. Then you have your inferior colliculus. Then you have your medial geniculate body. And then your auditory cortex. So E. coli MAC.